So for those of you who don't know me, my name is Natalie Fay. I'm an associate professor in the School of Chemistry at the University of Bristol. By training and conviction, I'm a computational chemist who does stuff with data. And that's really feeding into the motivation for us to organize this meeting. So this is a meeting jointly organized between the UK Catalysis Hub and RAW. There was a version of events in which we would have met in person and you would have been able to have a tour, as Ben was saying in his introduction to the Raw facility. But we decided that there was still so many uncertainties about travel, about COVID, and we were also able to access a much wider range of speakers, including some from the US. By keeping the meeting online. So what we've ended up with is 2 afternoons of talks. They are designed to highlight the impact of new technologies and approaches on catalysis and its link with data analysis and potentially going all the way to machine learning. As I was saying, it's a cooperation between the UK Catalysis Hub and RAW, and my colleagues Adrian Mulholland and Mimi He and myself were members of the Catalysis Hub steering group, and that was really what started to get us talking about actually speaking to the community and highlighting some of the experiences that people have already got in this area, and also some of the projects, and not just talking about the good things, but also some of the challenges with this type of work. So over the next couple of afternoons, we will have 4 sessions. The first session today will be on flow chemistry with 3 speakers, and then on digital chemistry and the things that are related to this later on in the afternoon. And then tomorrow we start out with a session on automation, and we finish off with a session on what we've loosely titled Industry 4.0. So where some of our colleagues from industry share their experiences with actually integrating these technologies. That isn't the only time you will hear from some of our industrial colleagues and collaborators. We've got some of them integrated in the sessions we are going to have over the course of today as well. So it is hopefully a good mix of presentations from practitioners. After each of the topic sessions, we are planning to have a panel discussion. And the idea behind doing this is that it will allow the panel members to share a bit more about their experiences, answer some of your questions, and also interact with each other. And certainly in other meetings, we found that this is quite a useful format for actually getting a bit of discussion and interactions going. This meeting and running everything is very much done by Corinne and Josie. And we will thank them many, many times over the course of today and tomorrow. But this is just the first time I really want to acknowledge that Josie Goodall and Corinne Anyika from the UK Catalysis Hub make this possible. They handle the speaker invitations, they run the Zoom session, they try desperately to keep us all on time. And they will also help with managing the questions throughout the sessions. So that pretty smoothly slides me into some of the housekeeping remarks as well. But before I do so, one of the things that somewhere got lost in translation between in-person meeting and online meeting is lunch today. So it's absolutely fine if you're enjoying a salad or something else whilst listening to these talks. As you can see, the current speaker is spotlighted and the rest of you are not particularly visible. It's the nature of the beast, and unfortunately, that we are aware that people will be drifting in and out for some of these sessions. To help with that, at least a little bit, the talks and the discussion will be recorded, and then Josie and Corinne will sort out sharing the presentations where the speakers are happy for this to happen. We're not planning on sharing the discussion sessions. They will be used to inform some of the grant application processes in the Catalysis Hub. But if there's anything you've got any questions about or anything like that, then please contact Josie and Corinne, and hopefully we can help with information where that is necessary. So what I'm going to do, and hopefully not offend the lords of Zoom, is try and share my screen. Am I having more luck than Ben? Can you see?
Yes.
Yeah,
Yes, we can see it.
Hooray. Okay, so it's not a universal problem, perhaps, but the session on flow well, it will be afternoon by then. It is now afternoon, so the session on flow will be chaired by Ben. There are 3 talks in it, and as you can see, we've allocated 30 minutes to those talks. So what we've asked the speakers to do is to aim to finish their talk within about 25 minutes, leaving us 5 minutes for questions. And what we'd like you to do is to pop these questions in the chat. And if it's a long and complicated question, then just put a note in it saying, I would like to ask a question, please. By doing this, we can actually allow people to ask their questions in the order that they've come in. And apologies in advance if we miss something. I'm sure you can make yourselves known. And then we will, as the session chairs, invite you to ask your questions. So please don't unmute unless you're invited to do so. For the panel discussion at the end of each topic, again, we're going to invite you to post some questions to the panel. And as the session chairs, we'll prioritize them and hopefully manage to get through quite a lot of them. And we are normally going to have 1 or 2 starting questions just to kick off the discussion for you. And then throughout all of this, I don't think I really have to say this, but I will do just so we're super clear. We'd like to ask you to be respectful, professional, and courteous in your conduct as well. So hopefully with careful management, this will all go very, very smoothly. The thing that is not going very smoothly is that I'm speaking more quickly than I normally would. So we're running a few minutes early. And I'm just wondering, Ben, whether you want to use that time to give a quick introduction to the flow session, and then we'll get going. Would that be okay?
Yeah, if we start early, we'll stay ahead of the clock.
Or we get to lunch a little bit earlier. So if that's okay with everybody, I'm going to bet on most people who want to be here being here, and allow Ben to go a little bit more slowly and begin to introduce the session on flow chemistry. So thank you very much to everybody for joining. A special thank you to Josie and Corinne, and also Raw for supporting this event administratively. And over to you, Ben, for the start of the first session.
Well, welcome. So yeah, our first session, we are going to be talking about, we've got 3 different talks covering various aspects of how flow chemistry can support catalysis. So I guess we'll start with hearing from Lee Edwards from GSK, and he's going to talk about some trickle bed flow reactors. And then we'll move on to Anna Slater from University of Liverpool. I guess, what are we saying? Developing tools to discover and optimize complex chemical systems. So interesting title. Can't be a bit. I can't really lead you in too much more on that. And then Mark Perititis from Cardiff is going to then deliver our 3rd talk of this session, more on digital chemistry as a robust tool for the design of heterogeneous catalysts for direct amination reactions. So to keep ahead of the clock, and I guess I'll also be quite brief with our introductions. I don't want to take away too much time from the speakers. But first up, Lee Edwards. So he is a GSK subject matter expert in continuous flow chemistry, photochemistry, and also electrochemistry. So right on target for all the things we do and we're interested in. Previously, Lee developed GSK's global photochemical platform, and now he's responsible for their gas liquid trickle bed flow chemistry work, which is what we're going to hear about today. And then over his 25-year career in industry, he started off at Sandoz Pharma and Novartis, went to Maybridge Chemical Company for a while, and then he's been at GSK since 2014. And yeah, we're really privileged to have Lee here today to talk about the work they're doing at GSK. So without further ado, I'll hand over to you, Lee, and hopefully your Zoom works better than mine. You're muted. Actually, one final note, actually. So Lee can't stay with us for the discussion session, the panel discussion later on. So if you do have any questions for Lee, we'll try and get through as many of them as we can right after the talk, but he needs to leave fairly sharpish. So some of you may miss out, sorry.
Can everyone hear me and can see my screen?
Yes, and yes.
Brilliant. Thank you for inviting me to talk, but unfortunately I've got to leave directly after this because I'm actually visiting equipment this week in Singapore. So I'm literally doing the talk and then getting on a flight. So without further ado, I can actually move my screen forward. Excellent. So within the GSK Flow Laboratories, we have a working envelope between -70 and 300 degrees, and between 1 and 200 bar. And for photochemistry, between 254 nanometers and 800 nanometers. So we've published a number of our workflows. So on organometallics, photochemistry, and what I'll be talking about today, hydrogenations. So I'm going to go quite quickly because I've got a number of slides to get through. So options for flow for hydrogenations, you can use tubular reactors for homogeneous and heterogeneous catalysis. I wouldn't recommend heterogeneous because you will block the reactor, but there has been a movement forward into coated static mixers at the moment. So Roche are using these at the moment with Oliver Kappa. There's also the Taylor cuvette reactors for homogeneous and heterogeneous catalysis as well. So we've published on using a homogeneous reaction of Taylor cuvette reactor, which forms solids over the duration of the reaction. So the Taylor cuvette reactors helps push the solids out of the reactors. You can also use continuous stir tank reactors. These are preferred by Pfizer and MSD. You can also use flooded bed reactors, which is the typical H-cube, and trickle bed reactors, which is what my talk will be on today. So to cover some basic flow terminologies, probably everyone's used of steady state. It's basically a process that doesn't change over time. The one that people don't generally know about is a state of control. This is what we do in pharma. We set a state of control, which means we set a set of parameters. When the reactor is in these parameters, we collect. When they're outside these parameters, we don't collect, we send to waste. So all steady state reactions will be in state of control, but not all state of control reactions will be at steady state. So this brings me on to pseudo steady state. So this is a parameter that is changing over time. And this is the case with trickle bed reactors because we get catalyst deactivation. So why is GSK interested in gas liquid transformation? So from a portfolio review, 30% of our portfolio has at least one gas liquid transformation in it. This can be broken down into homogeneous and heterogeneous reactions, with the bulk being in heterogeneous. So when you further break these down to look at the individual reactions in heterogeneous catalysis, the bulk of the work we do is nitro reductions or protectant group removal, and this is what we focus on. So the challenges with batch hydrogenations, they are basically a complex three-phase reaction system with poor heat and mass transfer, and they are particularly sensitive to scaling. So they require large amounts of compressed hydrogen. Our limiting operating pressure in batch is 5 bar. So if you consider a 2,000 litre vessel pressurized to 5 bar, that's a lot of hydrogen. They require manipulation of large amounts of catalytic powders, including filtration. And we have long cleaning times on batch vessels. It's a week for our operators to clean a vessel on 3 shifts. So they basically have to clean the vessels by hand. Conversely, when you move to flow, we get control of heat and mass transfer by the gas distribution. It's the simplest system for our engineers. We have high local catalyst load-ins for difficult transformations. The reactor sizes are a lot smaller, so that means we can go to higher pressure. So for us, that's 100 bar at 150 degrees. We run it pseudo-steady state. We get large amounts of catalyst utilization and improved quality. So if you look just here, so this is one of our batch hydrogenations with a air-sensitive amine. So it's been dropped out of the reaction vessel in an airbag, and it's slightly yellow. By the time you get to the filtration, it's, well, it looks black, but it's actually blue. Conversely, this is the flow hydrogenation. So when we sent the photo of this to the person who was doing this stage of chemistry, the response wasn't very polite, but it went along the lines of stop messing about and stop sending me photos of water until they came up and saw the process actually running. So we avoid catalyst handling for the filtrations, and trickle bed's been around since the 1930s. It's not a new technology, but it is for pharma. So when you think about trickle beds, everyone thinks of oil and gas and columns that big. That's not what pharma use. So for us, a typical API is between about 100 kilos and 4.5 ton a year. That's what we're looking at. So if we look at this Ehrenfeld plate reactor, it's a 1 mil chip reactor. So if we have a 10 volume process, so a volume process is nice and simple for people who work on plants. So if you take 1 gram, it goes into 10 mils of solvent. On plant, 1 kilo goes into 10 liters. Nice and simple. So you have a 10 mil process at 60 mils a minute to this chip reactor. It means you have a residence time of 1 second, which means your process is in 6 grams a minute, or just under 9 kilos a day, or 3 tons a year. So options for processes in pharma are, if you need to deliver a 500 kilo batch, you can either process 20 kilos a day, it takes you 25 days to process your batch, or 100 kilos a day, 5 days. Depends on the size of your reactors. Or if you're continually running, if you process between 5 and 20 kilos a day for 300 days a year, that's 1,500 to 6,000 kilos a year. So it's well within the parameters of what we're looking for production rates. So this has actually been used on pilot plant, which produced 70 kilos of material in the past. So this is a pilot plant reactor. Conversely, when you move to trickle bed reactors, the ID of the columns we're looking at are between 3 and 200 mil, and 200 mil being on the big side, it's about 8 inches. So what I would say about the Thales Nano family, these 2 reactors here are upflow reactors, and the maxi here can be up or downflow. So other reactors available are the HL FlowCat and the PAR reactors. So we've actually processed close to 80 grams a day on a FlowCat using this 12 millimeter column here, which is just under 2 kilos a day, or if you're looking at 300 days a year, processing over half a ton of material. The key thing for using the pharma-sized trickle bed reactors is actually the particle size of the catalyst. So you can't use batch powders because they're too small and you give a massive pressure drop in your trickle bed reactors. Conversely, you can't use industrial size catalysts because their particle size is between 1 millimeter to 10 centimeters. They physically do not fit in our reactors. So we have to go to a new particle size between 50 and 400 micron. So this brings us on to tube to particle size ratio. So GSK have set our particle size ratio as 15 to stop maldistribution in our trickle beds. Bayer and Lilly use 10, other energies use 20. So this gives us, if you're in a 3 millimeter column, we can use a maximum particle size of 200 micron. And conversely, when we move to a 6 millimeter column, we can move up to a 400 particle size. The caveat with this is, though, within pharma, we're material limited. So our start materials tend to be expensive, and we don't have hundreds of grams of material to work with. So For flow, we are material hungry. I can make medicinal chemists cry, and I make process chemists cry on the amount of material I require from them. For scale up, we use the Ergo equation to scale up. So we maintain the same fluid velocity and the same flow rate to catalyst mass ratio. So we basically scale our pressure drop. So instead of being long and thin, as a lot of people look at trickle bed reactors, we go short and fat. So the systems are very complex, and there's massive amounts of interactions within a trickle bed reaction. So one of the first things you set when the particle size actually sets your first parameter when as soon as you pack the bed. Things that you can then change on the fly are pressure, temperature, and flow rate, which affect loads of other things. And the one thing that is an independent parameter is catalyst deactivation. So we've deactivated catalysts by having the wrong gas flow rate or the wrong liquid flow rate. And we also see deactivation as well if we have the wrong temperature, which I'll go in later on. Other things that cause catalyst deactivation are listed here. There are many, which I am not going to go into in just a time. So catalyst suitable for flow. So we set a very strict criteria for our trickle beds. We look at particle size distribution. The distribution needs to be quite narrow, like this one, rather than this broad one here. We look at mechanical strength, which is around 1,000 bar is what we look for. We look at pressure drop, activity and selectivity to make sure the catalysts actually work. Deactivation of the catalysts, if they pass all that, they're suitable for our platform. Let's dig into that a bit more. So we use Mulven to look at the particle size distribution. So we want tight particle size distribution. We will then look at the mechanical strength of the catalysts, either individually as single particles. If they're around 200 micron or above, we will look at individual particles. Below 200 micron, we look at the bulk strength. So these are just some strength charts for single catalysts at 200 microns, and we're looking at about 1,000 bar. So this one passed on strength. So this is a demonstration of one that failed particle size distribution. So it's bimodal. Particle size are beautiful, spherical, what we look for, but there's fines in there. And when we spoke to the supplier of the support, they weren't willing to go back and re-sieve this material. So that was an instant fail for us. For radium nickel type catalysts, we use FBRM and stirring to assess particle size and mechanical strength, because the damage caused by a stirrer is a direct indication of the strength of the catalyst's backbone. So you can see with this radium nickel 2800 here, this was the first initial FBRM readout. Within an hour, we'd smashed up the catalyst to an unusable particle size. Whereas you look at this radium nickel 3111A, which is our favored radium nickel, we see no change in the particle size distribution over time. Most of the literature will tell you that you can't use slurry catalysts in packed bed. We can. So we look for pressure drop as well. So under this particular setup, we're looking for a pressure drop of less than 10. Above 10, we can scale it, but it becomes a lot more difficult. So we prefer catalysts with a pressure drop of less than 10 bar. We also look for compression of the catalyst as well. It's a nice simple system. All we do is pack in a small OmniFit column to 6.5 centimeters and flow water through at 10 mils a minute. Water is possibly the worst solvent you can ever use for a catalyst. It's viscous and it has no compressibility products really, so it's ideal for us. Right. So we also do extended pressure drop testings over 24 hours. And we also then do varying the flow rate to see what effect that has on the beds as well. So this is an example of a failed alumina support at 10 mils a minute. Basically over 25 hours, we got an increased pressure drop over time. So we do a lot of screening up front in an Endeavour before moving into an H-Cube to test activity and selectivity. So from our point of view, one of the reasons why we test it in the Endeavour is to look at where the catalysts actually go. So this is a powder catalyst, and one of the reasons why our cleaning on plant vessels takes so long is because we splash powder catalysts literally everywhere, and we're looking at the moment at 200 micron particle sizes to go into batch because our cleaning time and cycle time will be a lot less. So we use a modified H-Cube to generate a lot of our data to reduce the amount of material we actually use. The reason why it's a modified H-Cube is because it removes the electrochemical cell from it. So this stops us from having a wave of hydrogen generated. So when you're doing development work for a process, what you want is a steady amount of hydrogen coming through. So if you starve your catalyst, it's not ideal. So when it's like this, if you're at the bottom of the curve, you're starving your catalyst at the top of the curve, you've got the required amount of hydrogen. So we also have this tied into our automation system. So we collect data on our system for temperature, pressure, and flow rates of both liquid and gas appropriately every second. So we're incredibly data rich, as well as taking samples then for how the chemistry is performed. So I hold around 40 catalysts for flow from a variety of companies. So the challenges are, ideally we want spherical catalysts at 200 micron. Failing that, supports at 200 micron. A lot of the time there's no business case from catalyst suppliers to actually make that for us, but that is slowly changing. So as part of the Pharma Innovation Program Singapore, which is the collaboration between GSK, Merck, Pfizer, and Syngenta, this is now changing because we're all pushing for the same thing. And if there's any catalyst suppliers out there, I'd be happy to talk to you. So chemistry we run in trickle bed or in gas liquid. We run nitro reductions, we run oxime reductions, nitro reductions, tempo oxidations, air oxidations, and ammonia, a typical three-phase that we can work with. So I'll move on to a very quick case study. So we had a project that was having issues with a hydroxylamine buildup just here. So hydroxylamines are potentially explosive and genotoxin. So to give you an idea what a hydroxylamine can do to a pilot plant facility. So this is an accident from 1977 where there was a hydroxylamine buildup and then went to run away, which then detonated. Luckily, nobody was killed in this incident, but you can see the effect this has on a pilot plant. So despite screening a range of palladium and platinum catalysts, we still had an issue with hydroxylamine until we moved to rainy nickel, which then all the issues went away and we didn't see any hydroxylamine at all. So we generally don't use rainy nickel on plant because it's pyrophoric. Generally, you don't have an issue until you come to filter it. So you can just see the sparkler effect here. So there's three versions of what rainy nickel will do. The sparkler effect, the metal will just glow, or you actually have a flame. So there's some thermal imaging, which while doing this test, discovered I maxed out my thermal imaging camera at 280 degrees. The temperature probe that was actually in the rainy nickel hit 407 before I chickened out and put the fire out. And what we have learned is that a rainy nickel fire doesn't start below an oxygen content of 14%. So if we run a lot of our pilot plant reactors when we're using rainy nickel in glove boxes. So, and again, rainy nickel is not particularly nice. It's a carcinogen, a teratogen. So a lot of the issues we have with batch go away when we move to flow. So we pack the reactors in a water slurry into the reactors. We then wash away the water with solvent, do our chemistry, and at the end we can put the reactors back under water to drop out the material later. The other advantage of rainy nickel is the nickel, we can have a lot more parts per million versus palladium. So this is one of our longevity or deactivation studies for this particular process. So we had 750 mgs of rainy nickel 3111A in a H-cube cartridge, which is about the same size as HPLC file. And we processed that over 11.5 hours, which was 90 grams. So from this, we could work out that 1 kilo of our catalyst would process at 120, a minimum of 120 kilos on plant. So this is a demonstration of some poor deactivations. So in this particular one, this one is temperature related. So at 50 degrees, we get a very quick deactivation, as you can see here. But conversely, if you have this at about 75 degrees, you get a much slower deactivation. So this sort of condition I can scale, this one I can't. So we also look at what deactivates our catalyst. So getting your stoichiometry wrong of hydrogen, if you starve the bed, they deactivate. If you get your concentration of your solutions wrong and you put a wrong concentration down your bed, if it's too concentrated, they will deactivate. And regeneration of rainy nickel is incredibly difficult. We're still looking into this at the moment. So the key things is we must have 3.7 equivalents of hydrogen when we're using rainy nickel. So this is our HCl flowcat. So this is just after we just first got, and we modified it within about an hour of actually installing it. So we added an extra pressure gauge just here so we can get pressure drop across the system. A year later, it's looking a bit more different. So it's completely been modified again. We've added again an extra gauge this time on our liquid feed, because what we found is sometimes if you have a gas liquid mixer here, you can basically turn that into a spray dryer and you'll block that system there. So an extra gauge here helps us work out if we're blocking the gas and liquid lines. We added a heater cooler for our liquid feed, so we can either heat it or cool it, depending on what we want to do. Added thermal imaging so we can monitor the temperature here. So we're incredibly data rich on this system as well, because we have 4 temperature probes, 3 pressure sensors, and gas liquid flow sampling about every second. And then we move on to the actual PAT, so process analytical technology that we have on the system. So again, it's been updated again here. So we have an at-line HPLC sampler, which means we can either sample on the high or low pressure side of the reactor. We've added a H-Genie. We have a lunar probe here, which is a fiber optic temperature probe, measuring temperatures every 1 millimeter along its length, and an IR inline, so we can monitor quality coming out the far side as well. So moving on to our PAT. I'm not going to talk about at-line HPLC because everyone's seen a HPLC. So our IR serves 2 purposes. It's for reaction monitoring, for quality, and also for resonance time distribution as well. So we check that our beds are packed exactly the same each time. So this is a resonance time distribution before we've bedded down the bed. So it's bimodal, means we're getting channeling, or it's monomodal, but not completely packed down yet. So after we condition the bed, which basically means flowing gas and solvent at double the intended process conditions and at 80 bar to bed down that bed. So we have exactly the same pack every time we do this. So the lunar probe is a fiber optic temperature probe, which we use in both the lab and our pilot plant reactors. So when you run the temperature probe top to bottom of the bed, you encounter channeling, but it's a simple fix by moving the probe from bottom to top. And this is how we have our pilot plant reactors as well. So to give you an idea of what we actually see when we're using a lunar probe, we have roughly 200 data points for a 20 centimeter long probe, and we're collecting data every second. So data files on this for a typical run are around 10 gig. We also use thermal imaging as well. Either as stills or as live monitoring. We tend to use these on our lab units, so when we don't have the temperature probes in place, because we can monitor where the hotspot is on a reactor. So if you're looking at nitro reductions, it's an 80-degree adiabatic temperature rise at 0.3 molar. So you know exactly where on your bed the reaction's taking place. So we can also then use it to determine if heated or cooled feeds, where the reaction's actually taking place. So having a heated feed is really quite key to where the reaction takes place. So if you don't heat your feed and go in at room temperature, you're using the middle of your bed. If you move to a heated feed, only going to 35 degrees, the reaction's taking the top of your bed. So this video here is showing us processing 1.2 kilos of material over, it was 17 days of operation. When the hotspot moves to the bottom, that is us turning off the bed for the day. So we've processed approximately 1.2 kilos of material over 17 hours of actual operation, ignoring the startup and shutdown at the end of the days. On 43 grams of rainy nickel, which is about 3.8 weight% of material, which meant that we could process a minimum of another 4 kilos of material, but we didn't have it. So we are really light on material. I think I need to speed up a bit in the interest of time. So I'm just going to show the consistent profile of material coming out of the reactor. It's consistently what we want over 19 hours. And the other thing that we did notice is the change in pressure drop over time. So this is running on day one, starting up the next day. We get a pressure drop over time, which is something that we're still looking into at the moment to understand what this means for when we move to plant. So on plant, we would be probably running 24/7 for the entire campaign. So we may not possibly see that. So I'll know a bit more about that shortly. So again, quick GC of typical profile of what we were seeing from this reaction. So what we're currently investing in is in a new pilot plant in Singapore for gas liquid transformations. It was due commissioning in 2021, but COVID's slightly got in the way of that, and I'm hoping to be commissioning the unit in Q1 next year. So our pilot plant reactors are designed for operation of between 5 and 15 kilos a day at 100 bar and at 150 degrees to cover both rainy nickel and palladium and carbon catalysts to provide clinical material and process understanding. So we have a 1-inch, 1.5 inch, and 2-inch reactors. The 2-inch and the 1.5 inch have distribution plates. The 1-inch doesn't have them because you don't need one at that sort of scale. So for the gas liquid distribution, we looked at how we did the distribution. Our distribution heads are priority to us, and I won't be showing any pictures of those. But how we went about monitoring this is basically we had this device made, which has 19 holes in the bottom of it, all with the same volume, with 19 ports at the bottom. So if you get your distribution right, and when you collect the distribution at the bottom through all of them should have the same amount of liquid in them. So that's where we got to, but it took us some time. So we were on generation 4 distribution heads to get to where we were, including a lot of CFD design. So this is the pilot plant reactor, which I'm going to be seeing on Friday for the first time, rather than just a picture. So this is our 1.5 inch jacketed vessel. So then we have a number of filters to stop any breakthrough if rain nickel comes through, because the guys on pilot plant do not want to be handling rainy nickel at all. And this is all in a glove box as well. So this pulls out towards you and swings to the left so we can empty them as well in the glove box. So very short summary. Hopefully I've showed you that we have new structured catalysts designed for flow. We have rainy nickels designed for flow now. We have 3 reactors, 100 bar system capability for 5 to 15 kilos a day, and new thermal monitoring technologies, which are incredibly data rich. So our target is commission the pilot plant next year to do demonstration of a packed bed and performance of our new catalysts, and to apply it on projects, and to continue developing our catalyst chain. So that's one of our biggest drawbacks, is getting catalysts to go into this. So if you think about a palladium on carbon, I can get 200 of them. I'm struggling to get 10. So anybody who works on catalysts and has any, I'd be interested. So again, another pitch for me to get catalysts. And that leads me to thank everybody who's worked on the project, particularly Enerit, an outstanding engineer who's actually now working in Singapore. Shane, Heather, and Erin, who have done a lot of the catalyst work, which they did it for a lot longer than me. I should remember Amber as well has done that as well. And my collaborators at Merck, Pfizer, and Syngenta, and the University of A-Star, who we're working on 200 micron catalysts. I'm happy to take questions. I think I've come in on time as well.
Brilliant. Yeah, no, right on time. Thanks, Lee. Yeah, is there any questions from the audience? Okay, so we've got a question here from Adam Clayton. So what is the most viscous solvent that you have used in one of your developed trickle bed processes?
Well, NMP was not fun, but we've used it. So NMP.
NMP. If there's any other questions from the audience, please keep them coming. I've got a couple of questions. I was going to second what you've been saying about the need for catalysts, heterogeneous catalysts for the slightly larger particle size, because we struggle to find them. And I think usually we have to go and make them ourselves, palletize them and things. But it'd be much nicer if you could just buy them in and kind of get started with the synthesis much quicker. I was also fascinated by the work you were doing with the FBRM work, where you were using that as a quick test for the structural stability of the catalyst. So I was curious as to how, did you have to do much calibration on that, or is it one of those things that's very obvious when it's not working?
I would say it's very obvious, but I would say Enerit did all that work, and that was proper engineering work, and I managed to avoid that. I waited out for her. She did the rest, so there wasn't too many complaints.
Brilliant. Okay, Natalie has got a question for us here. So, you mentioned that these experiments are very data rich. Do you see opportunities from this data for more general analysis and prediction, or is it a bit too specific, or maybe too hard to capture and curate such data in the long term?
So we're actually trying to sort out our data capture. So we have lab manager systems that capture all the data for us. It's now we're looking at how we can mine the data long term. So one of the biggest problems is when you're using 5 computers is getting the data stamps correct across those 5 systems. And when you're trying to overlay everything, it is a nightmare. So getting the timestamps right at the moment and getting everything more integrated is where we're looking at. So we have a long-term automation plan, which is hopefully kicking off next year as well, to integrate everything. So hopefully coming soon. I'll be able to talk about it more hopefully next year.
Brilliant. Well, we'll get you back to talk about it then. I think Natalie's just commenting meta tagging of data is a big challenge, but it sounds good. Okay, I'm very aware of time, but I see we've got a question from Ian Fairlam. So he's saying they typically use 100 to 120 nanometer supported palladium nanoparticles, things like PVP nanoparticles. Do you have any idea, are those problematic from a toxicity perspective at those size?
You use the worst word you can, nano, because nanoparticles are, how do you prove they're not in your API? So that's one of the questions that we do get quite a lot, is if you're taking, how do you know? It's basically, nano is a bad word. Yeah,
and it's not enough just to show that you've got less than such and such level of palladium, because I guess it's so unknown, the toxicity of nanoparticles.
Yes. So even though palladium probably is a nanoparticle, but it's palladium, and as long as I don't use that word, I'm very happy.
Ian's just followed up with agglomerated palladium or clusters? I think the question of those equally problematic.
Clusters, no. Yeah, we have to be very careful about the wording, is what I would say at the moment. So
yeah, and I've just had the message. We're 1 p.m, so we need to crack on. So thank you, Lee. Good luck getting to Singapore, and yeah, thanks a million for your talk today.
Yeah.
Okay.
Thanks a lot, guys.